Right. We are now getting well and truly into the key processes of making proteins. So, we looked at transcription in last week's set of videos, which we'll recover again. And then we'll get into more depth in the next stage, which is called translation. Um, this week's set of slides will be proudly brought to you by the last animal in the household, Sandra, our retirement doggo. So this is a picture of Sandra shortly after we got her, and then another picture of Sandra at her birthday last year. So you might be able to tell that she's put on about 10 kilos between those two photos, and she looks a bit more sprightly. Sandra was mistreated when we first got her, weighed about 15 kilos, and has a bit of trauma in her background, so she's very, very scared of her food bowl and things like that. And once she attaches to her humans, she just loves affection and just will follow them everywhere. So you might even be able to see her in this video as we go across. That's her during a little yoga session late last year that we had out the back of my house. And we took her on a houseboat as well, which she loved. All right, so the pencil icon is back in these slides, but one thing to consider is that there is a lot of information you're going to be presented with throughout this presentation. It's okay to not know all of it by the end, but I guess you really want to try and make as effective notes as possible, understanding that you might need to reference this presentation in particular later down the track. On the left-hand side there, you'll see these are some terms that we've talked about previously, and on the right-hand side are some of the newer terms that you may not be familiar with so far that we will start to talk about through this video and in subsequent videos. But I guess it just gives you a bit of a picture as to how much information has been presented in even just the last 2 or 3 videos alone. All right, so we're going to look at now transcription again in some briefer details, and then look at translation and their connection. And we're going to talk about this new type of RNA as well, and we're going to look at all the differences between the different types of RNA. So there are 4 types of RNA which you're actually going to see throughout this topic. And I guess probably the only thing that you need to know is RNA is a temporary store of genetic information, or a storage of genetic information that's not found in the nucleus of our cells, but found elsewhere. Each has its own specific function and its own specific size. So we have the mRNA, which we talk about being that direct copy of our DNA that is transcribed, which we talked about last week. We have another one called ribosomal RNA, which is, you'll find out in a bit, is found in the ribosome, a subsection of one of our cells next to the endoplasmic reticulum, which is actually where the proteins are assembled. We have this microRNA, which we don't talk about quite often. I guess it's just you don't need to worry too much about it, but it does help to support a process called gene expression after we go through transcription. And we'll talk about gene expression in a few slides' time, and then a few weeks' time, and then transfer RNA. So the ones that I want you to pay particular attention in knowing and being able to differentiate between is 1, 2, and 4 in particular. All right, so when I use this term protein synthesis, I am talking about the creation of a protein. And so it's pretty much simplified in this picture here. We've got your original DNA. You go through the first part, which is the transcription, creating the mRNA strand. And then you go through the next part, which happens outside of the nucleus of the cell. That's called translation. And that's where we end up with the final product being a functional protein that is able to do a job for the body. So the concept of protein synthesis means creation of proteins. We will talk hopefully briefly about transcription, as I know we did a video on it last week, but if you want to skip past it because you're feeling pretty confident with it, please do so. So In this particular type starting point of protein synthesis, we are looking at where DNA is transcribed or copied into a working form of mRNA. So a temporary storage of genetic material that is a set sequence of nucleotides long that has the code for a functional protein. And how this works is there are a variety of what we call of enzymes that initiate these to occur. In this particular one here, we see RNA polymerase. And what that does is that helps to split apart the two strands of DNA. And then what happens is the RNA polymerase actually goes about being able to build the new mRNA strand. And it does that by using what we call the template strand. So the RNA polymerase creates a complementary strand to the template, as you can see in that second picture. You can see that blue line is being assembled on the bottom strand of the DNA. And we say that that assembles from the template strand. The coding strand, which is the top strand, is not the one that is copied, but it will contain the exact same information as our mRNA strand because it is the opposite of the template strand. And that's that importance of that complementary base pairing. So that blue strand we are creating, I will repeat, has the exact same sequence as the coding strand, but is opposite of the template strand. The only difference is because it is RNA, you will see that the coding strand difference between the blue and the black, the RNA and the DNA, is that you'll see wherever the T is, you actually see a U base. All right. All right, and here it is summarized into words. Oh, we've got a visitor in the corner, so you can see that's Trevor. So this enzyme called RNA polymerase unwinds the DNA and actually finds the point where the gene sequence is starting. So the exact point where it needs to. There will be a certain sequence of DNA where the RNA polymerase will specifically bind to that knows where that's the start point for this protein that we're trying to create. It then will start to synthesize or get free-floating nucleotides and actually assemble that new sequence of RNA. The code is copied, and so, like I said before, it is copied from the template and will match the coding strand of DNA. The only difference you will see between the coding and the RNA will be that difference in uracil and thymine. Uracil will bond with the adenine or be complementary to the adenine from the template strand. And then pretty much what happens is as the RNA polymerase builds the nucleotide sequence, the DNA zips back up behind it. What happens is we have stop codes, so stop sequences of DNA as well along that protein strand, so along that sequence of information on the DNA. And as soon as a stop sequence or a stop signal, basically a sequence of letters, is reached that says that's the end of the protein, the RNA polymerase actually is able to disconnect from the DNA, and then the mRNA strand is created and can move out to then get all of those exons and introns spliced. So we've just got the useful information done. Another useful image and a question prompt, which goes on to what I'm saying just before. As you can see, I've color-coded it to try and help you with thinking about that question in terms of what the RNA and the coding strand would have in common. The color code tries to give that away, and you can sort of see that as well comparing the template and the mRNA strand. All right, you might also, when we talk about transcription here, this term called transcription factors that help to support the building or the synthesis of the mRNA strand. All you need to think about when you hear that term is the word protein. So it's not just as simple as one enzyme being able to split apart the DNA strand and copy from start to finish. There are several different proteins and enzymes that are involved with all of this kind of process. I guess we just talk about RNA polymerase as a key one, but you might also hear that term transcription factor, and they're just other proteins that help support the RNA polymerase to be able to start to do its job. All right. We're getting to the end point of what transcription has to offer for us. And so, as we talked about in the last slides, you have your pre mRNA strand that is created or newly synthesized, and it's got all of the useful protein information, but it needs to get rid of all the non coding sections. All right. And there, remember, they were called the introns, the short sequences within that were non useful for the protein and its functionality. So we have this process called splicing, where all of those introns or non-useful segments of DNA or original segments of what was DNA is now RNA being cut out, and then you have a much shorter mRNA strand. And as you can see in the picture there, that's ready to go. That contains pretty much the exact coding for whatever the protein is to be created. So key thing from this slide, introns and exons are found in the original transcript, so the original pre-mRNA, the direct copy from the DNA sequence. What happens there is we actually splice or cut out all of the introns, and so then we are left with a mRNA strand that only contains the useful protein information, so only the exons. And think about that as exons being information that is going to exit the nucleus, and introns being information that is going to go back into the nucleus or never be able to leave the nucleus. All right, so now we get into this next and new concept here. So this concept, translation, is the second step in the sequence, and this happens at a new part of the cell called the ribosome. Technically, the ribosome is not an organelle, but I guess we can somewhat think of it as possessing similarities to an organelle as well as being a subcomponent of a cell. And so translation is the second but also final stage in what we are doing in protein synthesis. I should have written, I believe, where essentially what we're doing is we're using the copied information that we originally found and got from the DNA and actually going, now we have the information, let's mass produce proteins from that. And then they're able to go through the bloodstream and initiate change in the body in some way, shape, or form. All right. And I guess probably if you could summarize it in one sentence, translation is the process of building a polypeptide chain using a sequence of codons or useful information. All right. And when we talk about a polypeptide chain, we are talking about a polypeptide chain that will eventually become a fully functional protein. But it's building multiple polypeptide chains to create what was called a protein. So our first step, the mRNA strand moves from the nucleus and it goes through nuclear pores, so little spaces within the nucleus, and it goes to the cytoplasm, which is a big open space in the cell, and then to a structure within the cell called a ribosome. Ribosomes are found connected to the rough endoplasmic reticulum. And there are hundreds or if not thousands of them that are all connected there. So this protein synthesis can occur at any one of those ribosomes. And so our mRNA strand goes out, finds its way to the rough endoplasmic reticulum, and then that's where we see where the ribosomes are connected to it, and that's where this next sequence in events can occur. So a little bit of information just on what a ribosome is. Think about it as just sort of a substructure or a subcomponent of a cell, and think about it where the actual protein gets created. So for the protein to be created, there has to be some way that the ribosome knows how to do its job, and so it contains its own ribosomal RNA, so rRNA. Basically, this is just the instructions within the ribosome that tell it, you are able to create proteins. Here's the instruction manual for how to do that. So what happens is at the ribosome, the rRNA, which is just the instruction manual that is within the ribosome, helps to pull through the strand of mRNA through and actually then instruct all, basically, this new sequence as tRNA, which we'll talk about on the next slides, to start assembling. And so it reads it in these 3-letter sequences or 3 base sequences called codons. And so on our mRNA strand, every 3 bases that we see together actually represents what will become an amino acid. And so we call the ones on mRNA, we call those a codon. All right, that's a key term that you just want to remember and associate that with mRNA containing codon information. All right. And then what happens is we're going to get to the next slide and we're going to talk about anticodons, which match that, and then what the anticodons will bring. So, another form of RNA. So, transfer RNA is this next form of RNA that basically, as this messenger or mRNA is being pulled through the ribosome, and you get these 3-letter sequences read, which are called codons, what happens is they'll have another 3 letters that are opposite of that. And that's called an anticodon sequence. And so that whole base pairing rule that we talked about in weeks 1 and 2 still applies here. And so you can see in this particular picture here, you've got a sequence of AAA. And so the tRNA molecule, which is the transfer RNA molecule, another set of genetic information, is brought to the ribosome. And that contains a 3-letter sequence called an anticodon that is the opposite base pairing rule to what we have in the original mRNA. And so that's a really, really important thing that tRNA has, because the structure of tRNA also allows it to bring about or bring across with it an amino acid that matches that sequence. And so what we can see here is we can see that we've got the AAA codon sequence. We have the UUU anticodon sequence that is the tRNA molecule that will bond to that. And then that brings with it this particular amino acid that is attached to the tRNA molecule that will actually correspond with that corresponding sequence. And so that starts the combination up the top there in those pink circles of the assembly of what we call a peptide chain. And so that starts the building, and it's like building Lego blocks, but having the instruction manual to go, build this Lego block next because you have this sequence next. And so it's like only specific ones will fit and match, and then they'll bring about specifically to tell you which next block to build and connect to. So, again, tRNA is another form of nucleic acid, so it still is basically like genetic information. It brings about and it transfers the amino acid because it's connected to the specific amino acid that codon sequence has, and the anticodon sequence has. And it brings about that corresponding amino acid that will have that anticodon sequence. So each tRNA molecule is unique, I guess is probably a good thing to note, and will contain an anticodon sequence. And then what it will have is each tRNA molecule will have a specific amino acid that bonds to it. And then as that gets pulled through, that can start assembling those amino acids, which will eventually create the polypeptide, which will eventually create the protein, which will eventually be able to go about and do a job for your body. So some new terms that we've talked about here, just to recap. Codon is a 3-letter sequence of information that's found on the mRNA strand, and that 3-letter sequence will eventually correspond to a particular amino acid sequence that we're looking for. Anticodon is the opposite of the codon sequence, and that is what's found on the tRNA molecule. And then if we look up the top there, we know that every tRNA molecule will bring about a specific amino acid depending on what the anticodon-codon sequence match is. All right. So once we've started to build those amino acids together and they're joining together, we can then refer to the joining of those amino acids as now a peptide sequence. So once we get a few amino acids together, we can now call it a peptide chain. And then eventually, once we start to hit a long enough chain of all of these assembled amino acids, we can start to call it a polypeptide. What ends up happening as well is then we go from polypeptide, and then once we produce multiple polypeptides, they will then bond together to form what is a functional protein. All right, and I'm going to skip through to this one. And what you can see here is that amino acids will contain a three-letter code. And so we know that if we see a three-letter coding situation on the mRNA or the tRNA, we can actually go either keep it consistent and go, that means this, or flip it around because of the codon-anticodon relationship. And we understand that if we see a sequence of information, we can correspond that to actually this will produce a particular type of amino acid. And so if we look at the top left corner there, we can see that the sequence code TTT on when we're looking at DNA actually will eventually be able to create the amino acid of PHE. We can also see that the TTC sequence will create the same amino acid. So if you see that in the original DNA strand, we know that that actually codes for that particular amino acid. You can also see on this, say if you go to in the top row and look at the third one across, the TAA sequence or the TAG sequence within the DNA actually means stop. So that's that stop point of stop producing or stop synthesizing this new protein. So that is a stop marker for us to know that's actually where we need to stop. That's where the RNA polymerase will stop, and that's where the mRNA will disconnect in the original instance. But I guess one thing to look at here is just you can see that there are multiple combinations of three-letter codons that will be able to form the same type of amino acid. So when we get things like random mutations that occur within our body, sometimes we don't notice the effect of them. Because if we have a random mutation, say, of TTT to TTC, that will still result in the same amino acid being produced, which will in turn, once we get to the protein stage, mean that that will still be able to serve the same type of function. And why do we need to know all this sort of stuff? Is because amino acids, when they combine together and make peptides that eventually make polypeptides that make proteins, essentially, we actually use those proteins to help the body. So proteins are useful for things like growth, repair, causing change in the body, breaking down food. All right, so they are useful for every single function that our body has to do is as a response to these types of chemicals that we produce. All right, what I will do is I'll skip through these, but I do recommend that you pick one of these that makes the most sense to you, and then have that within your notes as well. And before we finish, I would probably just have a look at these 6 steps here to look at, does this make sense? Does this make the most sense out of all of what we've spoken about today? Because this is transcription to translation in what I hopefully think is 6 easy or shortened steps. And basically at the end of this presentation and through the next student understanding, there's a whole bunch of different pictures that show the same thing. I guess what you need to do is just determine, do any of them make sense? Do none of them make sense? Ask the question then of me to explain them to you. Or if one of them makes more sense than the other, use that as one to add to your notes. All right. And always, as always, watch all the videos that are available to you for this kind of stuff.